Assalamu alaikum viewers, welcome to my channel Genome Study. If you are new on my channel, please subscribe and press the bell icon so you will get the notification of all the new videos. But don't forget to like the video. As we know that in life science, the gene cloning and recombinant DNA technology is a very important, and it is a backbone of the study for research of our gene through which we can characterize its function. So I have decided to start a series of lectures or videos which includes all the steps from step 1 to the end, that how we can select the gene of our interest and how we can clone it and transform it and how we can characterize it. So this is the part 1, and in this video I will give you a brief introduction about each step and that what type of steps include in this series. So first we will have to selection of our gene, that how we can select our gene of interest, what are the steps involved in the gene of interest selection, that any gene that we are going to select for our project that are you doing in your ms or phd or whatsoever. So what are the steps include in the selection of your gene and how we can select. And then after that means after selection, then we need to take its sequence, how we can extract or what type of sequence we will extract it, and how we can design the primer for its cloning, and of how we can design that primer and how we can get it from the company and how we can dilute that primer, how much concentration of it. And then we have to check it that these primer are work 100% or not, how we can check it. And then we have to need the RNA and cDNA for the cloning of that gene, and how we can isolate the RNA from the samples, that is the plant samples. If we are working, if you are working on the animal or any other, so you can get the RNA from that your respective organism, and then how we can synthesize that cDNA and how we can use it. And then we also do the qPCR and data analysis for further analysis of the selection of our gene, because that selection includes different steps. Then we have, after the confirmation and selection of that gene, then we will do the PCR for gene cloning, that how we can do that PCR and what type of PCR we can do for that the cloning. And after that PCR, how we can make the gel and how we can run that gel and how we can analyze our gene and how we can that cut and purify that gel to extract our gene. And then we can do the enzyme digestion, that how what type of enzymes we have to use for the digestion of our gene, and also the vector that we are using for that cloning. And then we can use the ligation, that how we can ligate that our gene into the TZ vector or any other vector. And then after that means its gene of interest ligation in another vector that we have to use in our studies. For example, if we want to overexpress that gene, that we need a overexpression vector. If we knock down the gene, that we need RNAi. Or if we use to do the protein assay, then we use the related vectors. So other vectors also, how we can ligate our gene in that vectors, and then how we can transform it into the E. coli, and which strain of E. coli we have to use, what are the protocol for the transformation, and then how we can select that clones, and how we can confirm that clones are positive clones and that clones contains our genes that clone is. And how we can then we have to send for the sequencing of that clone is, that of E. coli that we have selected after different steps. And then we have to confirm through sequencing that our gene of interest was inserted or located in our respective vector. After that, we have to transform it into different organisms like for a overexpression of RNA, or for the transformation in the plant, we have to transform it into the agrobacterium strains like rhizogenes or tumefacients according to our purpose, or we have to transform it into the yeast for further analysis. How we can, or which method we have to use, how we can do that one, and then how we can also transform into yeast and yeast assay analysis, and then how we can do the transformation in Arabidopsis as a model plant or in our respective plants through that agrobacterium, and then how we can select that Arabidopsis seeds, transform seeds, and then when do what type of gene analysis we have to do in Arabidopsis, and then cell localization for also the characterization of that gene involved. Then we have to do the WGCN analysis to find out that what type of interactive genes or transcription factors were involved that have the interaction with that our gene of interest, and how we can do further analysis for the confirmation like the yeast 1 hybrid and yeast 2 hybrid, and further some other analysis. So all these steps will included in this series of lectures. If you have any other idea or any other experiment that you need that we have must included in it, so you can also comment it, and hopefully this series of lecture is very useful for you all. If you have any query, please comment and don't forget to like. Thank you.